Welcome to the Longevity Papers podcast.
Glad to be here.
It is September 2nd, 2025, and today we are embarking on, well, our regular look at the latest research, specifically curated for you, the biotech researcher. We know you're probably swimming in abstracts, right? Trying to figure out what actually moves the needle, what deserves your precious time in the lab,
Yeah.
and what genuinely shifts our understanding in this incredibly dynamic field of longevity. It's a lot to keep up with.
It really is. And let's be honest, from the outset, as you probably find most weeks, longevity research tends to bring incremental advances, not really huge paradigm shifts week after week.
Right.
So our job here, as we see it, is to help you distinguish between the two, providing you with a clear lens and, frankly, a healthy dose of skepticism.
Which is so necessary.
It's probably your most valuable tool in navigating this landscape, which, let's face it, is often full of hype.
Absolutely. Our mission is to extract those truly important nuggets of insight from the constant fire hose of information, offering you a shortcut to being genuinely well-informed. We want to help you understand the so what for your work, your grant applications, or just your core interests in longevity. We are here to be, truth-seeking and honest with you today. And the truth is, there are no truly amazing model-changing papers that just landed this week that are going to suddenly redefine your entire research direction.
That's the reality most weeks.
But there are some interesting threads worth pulling on, some significant movements maybe in the broader landscape, and as always, some crucial lessons in critical thinking.
And that's such an important distinction to make. True breakthroughs are rare, but it's often in these seemingly smaller steps, the incremental refinements, the novel methodologies, maybe challenging existing assumptions, that the foundation for future, truly disruptive breakthroughs is laid.
Right. You build on those smaller steps.
Exactly. And understanding their limitations is just as important as appreciating their potential. You need both sides.
Okay, so before we dig into what hit the preprint servers and journals literally this week, let's zoom out a bit first.
Good idea.
We just wrapped up the ARDD, right? The Aging Research Drug Discovery Conference in Copenhagen last week.
Always a big one.
Yeah, it's always a huge barometer for where the field is, or at least maybe where it's willing to publicly discuss going. Historically, ARDD tends to shy away from what some might consider the more radical or audacious approaches.
That's generally true, yes. They tend to be more focused on, say, druggable targets.
So what stood out for you from the conference this year? What kind of emerging trends or, shifts in the conversation did you pick up on?
Well, what was really striking this year, I thought, was a palpable shift in the nature of the discourse. For the very first time in ARD's history, there was actually a dedicated replacement panel.
A replacement panel? Wow.
Yeah. This is a considerable departure from the conference's usual, often more conservative tone, focusing on drug targets and established mechanisms. It signifies, I think, a growing willingness within the mainstream longevity community to seriously discuss interventions that are far more audacious than just, drug discovery or cellular repair or even partial reprogramming. It implies a move towards a kind of if it's broken, swap it out mentality, but on a much, much grander scale.
That's a monumental conceptual leap, isn't it? It feels like we're moving from trying to meticulously patch up an old ship.
Yeah, exactly.
To discussing building entirely new modules, or maybe even a new ship around the old one. Who were some of the key figures driving that discussion? And what was the general feeling like? Was it excitement, skepticism?
Well, it's fascinating reframing of the challenge of aging, for sure. The panel was hosted very well by Sierra Lohr, and among the prominent figures was Vadim Gladyshev.
Ah, from Harvard.
That's right. A renowned professor of medicine from Harvard Medical School, known for his work on molecular mechanisms of aging. Gladyshev, in particular, made a pretty bold prediction during his session.
Oh, what did he say?
He stated, quite definitively, that full-body replacement is likely to happen by 2100.
Whoa, full body replacement by 2100.
Now, that's a statement that certainly got everyone talking and definitely turns heads, beyond the typical longevity circles.
Yeah, no kidding. That does sound straight out of William Gibson or Philip K. Dick.
It does a bit, doesn't it?
But if someone of Vadim Gladishev's stature is putting that idea out there at a serious scientific conference, well, there must be some deep underlying thought and perhaps even an emerging theoretical framework behind it, right?
Absolutely.
Does this audacious claim connect to anything we've seen in the scientific literature recently? Is it just a conference soundbite, or is there some real substance there.
No, it absolutely connects, and you're right to question if it's merely speculative.
Mm.
This isn't just a new, spontaneous idea dropped at a conference.
Yeah.
Gladyshev is actually a co-author on a pretty seminal perspective paper titled Replacement as an Aging Intervention. It was led by Sierra Lohr et al., and as a matter of fact, it was published 4 months back.
Ah, okay, so it's been out for a bit.
Yeah, May 8th, 2025, in Nature Aging. And it's important to note, our own longevitypapers.com site actually tracked that paper when it first came out.
Right.
We recognize its significance even then
Mm.
as a signal of, shifting perspectives in the field.
Ah, so this idea has been brewing and has already landed in a top-tier journal. That definitely provides a lot more context and weight to the ARDD discussion. Okay, so let's dig into this paper a bit then.
Sure.
What's the core argument of Lohr et al.? Why are they advocating for replacement as an aging intervention when so much of the field is intensely focused on, slowing down or reversing aging processes directly? At the cellular level, the molecular level. It almost feels like giving up on fixing the original parts.
Yeah, I can see why it might feel that way. But I don't think it's so much a surrender as it is a pragmatic reevaluation of where we are right now and maybe looking back at historical successes in medicine.
Okay.
The paper makes a very compelling argument by highlighting a critical disconnect. While we've made substantial, undeniable progress in understanding the fundamental mechanisms of aging, telomeres, senescence, mitochondria, epigenetics, all that.
Right. Hallmarks.
Exactly. There's still a noticeable, frankly, sometimes frustrating lack of conclusive human interventions that have been unequivocally shown to genuinely attenuate or reverse the aging process in a holistic, clinically meaningful way. We have promising leads, lots of them, but no definitive cures for aging itself yet. In stark contrast, though, if you look at the history and current practice of modern medicine, replacement interventions have a very long and undeniably successful track record.
Okay. Like what?
Well, think about it. Joint replacements for arthritis restore mobility dramatically. Pacemakers completely take over the function of a failing natural pacemaker. Or consider transplant therapies replacing a diseased kidney or liver or heart can massively extend and improve a patient's life.
True. Very true.
These approaches have been incredibly effective at restoring function when you have acute injury or chronic disease. So the paper logically posits, why can't we apply this same robust logic, these proven replacement strategies, as interventions specifically for the diseases and dysfunctions of aging itself? It's about leveraging existing medical successes within a new conceptual framework for longevity.
That's a truly fascinating reframing of the problem. Instead of trying to painstakingly fix every single tiny aspect of senescence or protein misfolding or mitochondrial dysfunction in an old tissue,
Right.
why not just, as you said, replace the entire worn out part with a new functional one? It's a bold thought. So what kind of strategies does the paper actually discuss? Are we just talking about swapping out organs, or is it something much more advanced, maybe cellular or even synthetic?
Oh, it's a much broader and more nuanced discussion than just simple organ swaps. It really pushes the boundaries of what we currently imagine.
Yeah.
The paper outlines several strategic categories for these replacement interventions.
Okay.
On the biological sides, they discuss cutting-edge innovations in tissue engineering, using sophisticated biocompatible scaffolds, advanced bioprinting techniques to generate functional tissues, or even complex organs de novo, basically building new biological parts from scratch in the lab.
Right, like 3D printing an organ?
Kind of, yeah. Then there's genetic engineering, focusing on enhancing donor-recipient compatibility, which is a huge hurdle in current transplants, right? Often requiring lifelong immunosuppression. They also delve into cell therapies, where specific cell populations could be replaced or rejuvenated, and xenotransplantation strategies, using animal organs, like from pigs, modified to be compatible with human recipients.
Okay, so those are all about creating or adapting new biological components.
Exactly.
But what about synthetic approaches? Are we talking about like super advanced prosthetics or something even more integrated?
Precisely. On the synthetic front, they explore advanced prostheses way beyond what we consider today, moving towards truly integrated functional replacements for limbs or maybe even other body parts. They also consider external devices, like advanced circulatory support systems or external artificial organs that could sustain or enhance function outside the body. And perhaps most audaciously, they discuss brain-machine interfaces. And these aren't just about controlling a prosthetic arm, They envision interfaces that could potentially replace or augment neural functions, maybe addressing age-related cognitive decline or even memory loss. These are all ways to restore or enhance function using non-biological components that could, theoretically, seamlessly integrate with the body. And what's particularly fascinating here, I think, is that they also evaluate existing evidence for systemic benefits that might extend beyond just the local replacement.
What do you mean by systemic?
Well, they point to the well-known heterochronic parabiosis experiments in mice.
Right, linking old and young mice circulation.
Exactly, where connecting the circulatory systems can lead to systemic rejuvenation effects in the older mouse, implying that young blood factors can have broad positive effects. They also look at human data from donor-recipient age mismatched transplants, where a younger organ put into an older recipient often performs better than an age-matched one would have. This suggests that the local environment of an aged body doesn't completely negate the benefits of a younger, healthier component. It implies replacement could have cascading positive effects on the rest of the system.
Okay, so this isn't just about replacing a single faulty part like a worn-out car tire.
Right.
It's about the potential for these replacements to trigger broader rejuvenating effects within the body, perhaps even restoring a more youthful systemic environment. That's a truly powerful and intriguing idea. It shifts the focus from just individual organ failure to maybe systemic rejuvenation through strategic replacement.
It really has.
But I can't imagine this comes without very significant hurdles, right? Both scientifically and societally. What key challenges do Lore et al. Actually highlight in their paper? They can't just be presenting this as a seamless future.
Oh, absolutely not.
Yeah.
You're right. They are very realistic and forthright about the monumental challenges ahead. Ethical considerations are paramount, naturally, especially as we move toward more complex or widespread replacements.
Yeah.
Questions about identity, consciousness, the very definition of human, and crucially equitable access, who gets these potentially life-extending technologies, would become incredibly pressing.
Huge question.
Huge. Beyond that, scalability is another massive practical concern. How do you possibly produce enough bioengineered tissues or compatible synthetic parts, or find enough donors for a global population that might eventually need such interventions? It's not a small feat.
No.
And of course, they discuss the vast future directions required in research and technological development to even advance these replacement therapies to become truly viable, safe, scalable, and importantly, ethically justifiable interventions for aging. We're talking about fundamental advances needed across multiple scientific and engineering disciplines.
So drilling down into this, the shift towards even discussing full body replacement at a major conference like ARDD and having this comprehensive paper outlining the scientific and engineering landscape, well, it's a truly fascinating signal about where the field might be headed, even if it's many, many decades away. It really forces us to consider a much wider range of potential solutions to aging, not just molecular tweaking, but audacious bioengineering. It's a conceptual framework shift that's kind of hard to ignore.
It absolutely is a conceptual shift.
Yep.
It asks us to consider whether continuously patching up a deteriorating system is ultimately less efficient, maybe, than strategically replacing its core components with new ones.
Yeah.
And speaking of broader trends from ARDD that kind of contrast with this long-term audacious vision,
Yeah.
another significant development we saw was the increased pharma presence this year. Giants like Novo Nordisk and Eli Lilly were very visible.
Ah, the GLP-1 players.
Exactly. Making some significant claims, particularly regarding GLP-1s, essentially positioning them as a potential longevity drug. It's an interesting development, and it speaks to the commercial interest and the rapid translation of certain metabolic therapies into the longevity discussion. But it's important to keep that in context with the more fundamental and, in some cases, more speculative research we've just discussed. It's sort of the short-term clinical application versus the very long-term vision.
Absolutely. The commercial buzz and immediate market opportunities are one thing, but the deep scientific exploration, even the speculative, potentially groundbreaking theoretical work, is what truly drives long-term transformative change. Okay, that was a truly insightful glimpse into the cutting edge of thinking, some of the very audacious long-term visions being discussed at the highest levels. Now, let's bring it back to what actually landed on our desks this week.
Back to the present.
The latest papers for you, the biotech researcher, and as we said at the top, we need to calibrate our expectations carefully for the weekly grind.
Indeed. This is the reality of scientific progress, right? With about a 95% probability, any given week will contain only incremental advances. Things that are more likely statistical fluctuations or maybe refinements within existing paradigms than genuine paradigm shifts.
Yeah.
It's vital to recognize that because it prevents overreaction to every new abstract, which can lead to wasted resources and misclick focus in the lab. But there are still valuable lessons in these smaller steps. Lessons that refine our understanding, push methodologies forward, and sometimes even provide a crucial piece of the foundation for that future model-changing work. So let's dive into these with the critical lens they deserve.
Okay, shifting gears from speculative futures to the cutting edge of the weekly literature, our first deep dive takes us into something that always sparks interest. Yamanaka factors.
Always a hot topic.
Yeah, this one caught my eye because it involves those reprogramming factors. It's titled Reprogramming Factors Activate a Non-Canonical Oxidative Resilience Pathway that can rejuvenate RPEs and restore vision.
Right. This study, emerging from the Whitehead Institute, which is obviously a top institution,
Mm-hmm.
represents one of the more interesting findings this week, I'd say. Yet it absolutely requires significant skepticism from you as a researcher. It's a Bayer H preprint, published just yesterday, September 1st, 2025.
Okay.
And for those unfamiliar, Bayer H is pronounced bioarchive, B-I-O archive. It's a repository for preprints before they go through formal peer review.
Good to clarify that pronunciation, bioarchive. And for those who want to look it up, the primary authors are Lou, Y.R. Cameron, J.C., and Hugh Wywick. We'll link it, of course. But so what are the key claims coming out of their abstracts, specifically regarding these Yamanaka factors and vision?
Okay, so the core claim builds on established knowledge, right? The Noctami-4, SOX2, and KLF4, often abbreviated OSK, the canonical Yamanaka factors can induce pluripotency and reverse age-related epigenetic changes. That's fairly standard ground.
Right.
However, the true novelty they're presenting is a specific, targeted mechanism. They find that OSK expression, when carefully controlled within the retinal pigmented epithelium, or RPE.
The RPE in the eye.
Exactly. It not only restores retinal structure, but also significantly improves visual function in aged mice. And that's a very compelling and specific functional outcome.
That's a pretty bold claim, not just, making cells look younger under a microscope, but actual restoration of visual function. How are they saying this dramatic improvement is achieved? What's the underlying mechanistic pathway they've identified?
They posit that this is achieved by promoting oxidative resilience in the RPE cells.
Yeah.
But crucially, they say it's through what they describe as a non-canonical TET2 independent pathway.
Okay, non-canonical and TET2 independent. Why is that distinction important?
It's important because it suggests a different route than some previously understood reprogramming mechanisms that involve the TET methylcytosine dioxygenases, which are key epigenetic modifiers. Essentially, they're saying OSK isn't just broadly resetting the epigenome here. It seems to be triggering a very specific protective response pathway.
And did they identify the key player in that pathway?
Yes. Through intensive integrative functional genomics looking at gene expression changes and interactions, they pinpoint GSTA4, that's glutathione S-transferase A4.
GSTA4, I know that one. It's a detoxifying enzyme, right? Clears lipid peroxidation products like 4-HNE.
Exactly that one, a well-known detoxifying enzyme. They identify it as a necessary and sufficient effector of OSK in this particular process. They're saying GSTA4 is the key downstream player doing the heavy lifting for this oxidative resilience.
So GSTA4, a specific detoxifying enzyme, is the star of the show here, a critical downstream effector of OSK in this context. That is interesting. What are the broader implications the paper suggests from this discovery? Do they think it extends beyond just eye health?
Well, the paper goes on to make a series of quite ambitious claims based on this. First, they assert that this dynamic GSTA4 regulation by OSK effectively recapitulates a kind of stem cell-derived stress resilience program, implying it's tapping into a fundamental protective mechanism.
Okay.
Furthermore, they claim that GSTA4 overexpression alone, without needing the full suite of OSK factors, is sufficient to enhance mitochondrial resilience, rejuvenate the aged RPE transcriptome, basically, making the gene expression patterns look younger, and, perhaps most importantly, reverse the visual decline they observed.
Wow, GSTA4 alone doing all that.
That's the claim. And perhaps most ambitiously, extending beyond the eye, the paper claims that GSTA4 is consistently upregulated across diverse lifespan-extending interventions. They suggest it might have a much broader pro-longevity role, acting as a general mechanism of protection against oxidative stress across various tissues.
Okay, here's where it gets really interesting for our listeners, right?
Hmm.
The idea that OSK could restore vision and rejuvenate RPEs without full reprogramming by leveraging a specific effector like GSTA4, that's incredibly compelling for anyone in biotech aiming for targeted, safer therapies.
Hmm.
Because for many, the word Yamanaka still carries the baggage of full pluripotency and, tumor risk.
Absolutely. Teratoma risk is the classic concern.
So how convinced should we be that this non-canonical pathway truly sidesteps those fundamental safety concerns? Is it really a safer route or just a nuanced risk reduction? And what are the other crucial points a researcher like you needs to consider before getting too excited about immediate translation?
That's an excellent critical question. And it's exactly where the skepticism needs to kick in hard. While intriguing, this is, at its core, an incremental advance. It builds on the well-established knowledge that Yamanaka factors can reverse aging markers. The key novelty here, for you, the researcher, is identifying GSTA4 as the critical effector and proposing this non-canonical pathway.
Right.
However, before you dedicate significant resources based on this preprint, several crucial points need very careful consideration. First, and absolutely paramount, is independent replication.
Always number one.
Always. Visual function is a complex, often subjective phenotype in animal models. Different lighting conditions, different behavioral assays, even the strain of mice used can introduce variability. These kinds of functional findings need robust verification across multiple independent labs using diverse methodologies to confirm the effects aren't just due to subtle experimental biases or, frankly, statistical flukes.
Makes sense. What else?
Secondly, we really need a clear delineation of dose-response relationships. The paper needs to precisely define the optimal levels of OSK expression and, even more critically, how GSTA4 expression levels relate to the rejuvenation and vision restoration they observed.
Like, is there a sweet spot? Too much or too little is bad.
Exactly. Is there a therapeutic window? What are the potential consequences of over or under expression? Without that precision, it's hard to understand the true therapeutic potential or, crucially, the safety margins. And to your point about safety, even if they're bypassing full pluripotency, what are the long-term safety data?
Mm-hmm.
We need to know what happens months or years down the line with OSK expression, even in this supposedly non-reprogramming context. Partial reprogramming still carries risks, potential off-target epigenetic changes, immune responses, unintended cellular phenotypes that might only show up later. Bypassing full pluripotency doesn't automatically mean bypassing all safety concerns, particularly the oncogenic transformation risks, even if they might be reduced.
That's a really critical safety point for any potential therapy. What about the broader applicability of this pathway? You mentioned they claim GSTA4 is involved in other longevity interventions. Could this be a general strategy, or is it maybe highly specific to the RPE?
That's precisely the translational question, isn't it? We need to ask if this translates effectively beyond this specific RPE model. Could this oxidative resilience pathway, driven by GSTA4, actually be leveraged in other tissues or organs that are particularly prone to age-related oxidative stress and dysfunction?
Like the brain or muscle?
Exactly. Brain, muscle, liver, kidney. That would significantly increase its impact and potential as a generalized longevity intervention. And regarding their broad claim that GSTA4 is consistently upregulated across diverse lifespan-extending interventions, well, that needs rigorous, unbiased validation.
Oh, so?
It could easily be cherry-picked data, right? Or maybe just a statistical artifact from looking at a few specific examples. What we really need is a systematic meta-analysis across a wide range of well-characterized interventions and species, looking for consistent upregulation patterns, not just a suggestive statement pulled from a few convenient studies. You, as the researcher, need to scrutinize that data yourself, not just accept the assertion in the abstract.
Right. Don't take the claim at face value. Check the underlying evidence rigorously. Are there any other alternative, perhaps simpler explanations for the vision restoration they're observing? Could it be something other than this specific pathway?
Absolutely. Vision restoration is a complex physiological outcome, especially in an aged system. There could potentially be multiple confounding explanations. For example, maybe OSK expression has general anti-inflammatory effects in the eye, which are known to improve RPE health. Or perhaps there are metabolic shifts induced by OSK that benefit the RPE indirectly, or even indirect trophic support to the RPEs coming from other nearby cell types that were affected. Any of these could impact RPE health and visual function without GSTA4 being the singular critical effector they claim it is.
So you need controls to rule those out.
Exactly. Careful control experiments are needed to really disentangle these possibilities. And finally, the big question. Does this genuinely require a model change in how we think about rejuvenation? Or is it really a novel, perhaps important detail within the existing paradigm of partial reprogramming?
And your take?
For now, I'd argue it's the latter. It refines our understanding, providing a specific, potentially actionable target like GSTA4 within the broader concept of partial reprogramming. But it doesn't fundamentally overturn the approach of using Yamanaka factors for rejuvenation. It adds a layer of mechanistic detail.
Okay, so what's the big idea here for you, the biotech researcher listening? It sounds like it's a promising lead, offering a specific testable mechanism, GSTA4, that could be incredibly valuable for targeted therapies and ocular aging, maybe paving the way for more precise and safer interventions than just throwing the whole OSK cocktail at cells.
Hmm.
However, we're clearly a long way from an aha moment that changes clinical practice. It feels like a foundation for a lot more work, not the finished building.
That's a good way to put it.
The key insight here maybe is, while partial reprogramming holds immense promise, its true power might lie not in broad systemic resets, but in uncovering and targeting specific effector pathways like this GSTA4 one for precise, localized tissue rejuvenation, hopefully minimizing those systemic risks.
Exactly. It's a move, perhaps, from thinking about a broad-spectrum antibiotic approach to cellular repair towards more of a surgical laser.
I like that analogy, surgical laser.
Yeah. It provides new avenues for investigation and validation, pushing the boundaries of what partial reprogramming might achieve. But the critical work of verification, safety assessment, and broader mechanistic understanding is definitely still ahead. It's about building a robust, targeted therapy, not just demonstrating an interesting biological effect in a mouse model.
Right. Got it. Okay, next up, we have a study that touches on something many of you are intensely curious about, and perhaps even practicing yourselves: lifestyle interventions, specifically their direct molecular impact on aging.
Always relevant.
The title is Multimodal Atlas of Lifestyle Interventions Reveals Malleability of Aging-Linked Molecular Features.
Right. This University of Innsbruck study provides a truly comprehensive multi-omics atlas of lifestyle interventions. It's an impressive methodological undertaking just in terms of the sheer scope and the amount of data generated.
And where did this one appear?
This is another biography preprint, also published very recently on September 1st, 2025. So hot off the virtual press, also awaiting peer review.
Okay. And the authors for this one are Herzog, CMS, Vavarakis, CD, and Rettel E. We'll link that one too. So what is this Tirolges Lifestyle Atlas, and what exactly did this ambitious study involve? Who participated? What did they do?
Okay, so the paper introduces the Tirolges Lifestyle Atlas. It's registered on clinicaltrials.gov, which is good practice, under NCT 05678426. It aims to be a fantastic resource for the community. The study design itself is quite robust for a lifestyle intervention study. They profiled a cohort of 156 healthy women aged between 30 and 60 years old.
Okay, healthy women 30-60. What were the interventions?
These participants underwent one of two 6-month interventions. The larger group, 114 of them, engaged in an intermittent fasting regimen. The other group, 42 women, were part of a smoking cessation program. So this allows for a comparison of different types of impactful lifestyle changes on molecular markers.
That's a decent sample size for a human intervention study, especially one promising detailed data collection. How comprehensive was that data collection? What kind of multimodal information did they actually gather?
This is really where the multimodal aspect shines and sets this study apart as a potentially valuable resource. Participants were sampled extensively, up to 4 times over the 6-month intervention period. And crucially, they sampled across 7 different tissues and fluids.
7? Like what? Blood and...
Yeah, blood, urine, saliva, stool, and even things like skin biopsies. This generated an astounding data set of over 3,450 biospecimens in total.
Wow.
And for each of these, where appropriate, they performed harmonized DNA methylation profiling. For insights into epigenetic age clocks and changes, they did metabolomics for a snapshot of metabolic pathways, microbiome analysis from the stool samples, detailed immune profiling from blood samples, skin histology and barrier measurements from the biopsies. And on top of all that, they collected a rich set of clinical metadata, anthropometrics, diet questionnaires, physical activity logs.
That is truly a rich, unparalleled data set. It provides an incredible longitudinal view of how lifestyle changes affect molecular systems across different parts of the body.
Absolutely. It's an impressive undertaking from a data generation perspective.
So how did they actually demonstrate the utility of this massive data set? Did they just dump the data, or did they perform analyses to extract insight?
They definitely put the data through its paces to showcase its potential. They used it to perform multi-omics-wide association studies, or MWAS, linking various clinical traits and lifestyle factors to specific molecular features across different omics layers.
Okay.
They also employed integrative factor modeling to try and reveal coordinated cross-tissue molecular signatures that respond to the interventions. They looked at epigenetic biomarker cross-omic associations to find robust links, say, between methylation changes and metabolite levels or immune cell counts.
Hmm.
Furthermore, they conducted something called CpG-level variance decomposition. This basically allowed them to precisely pinpoint how much the variability in methylation patterns at specific sites
Hmm.
in different tissues is driven by individual genetics versus the specific tissue type versus the lifestyle intervention itself.
So disentangling the different influences.
Exactly. It's a powerful way to dissect the various influences on epigenetic aging markers. Essentially, they showcased its utility for uncovering complex biological relationships and identifying potentially robust biomarkers of response to these interventions.
That sounds like a fantastic resource for hypothesis generation and maybe biomarker discovery down the line. But now for the moment of truth, what's their core claim? What's the big takeaway for aging that they pulled from all this analysis?
Okay, their core claim, derived from this extensive data set, is, and I quote, aging-linked features are selectively malleable.
Selectively malleable. Okay.
More specifically, they state that highly compliant intermittent fasting participants exhibited attenuated or even age-opposing molecular trajectories within 6 months.
Age-opposing trajectories? That sounds like reversal.
That's certainly the implication. They observed changes in DNA methylation patterns, certain metabolites and immune cell populations that shifted in a direction generally considered more youthful based on established aging clocks and markers. And importantly, they've made this data available via an interactive portal with sensitive data under controlled access, which is an excellent contribution to the research community for validation and further exploration by others.
Wow. Age-opposing molecular trajectories from lifestyle changes in just 6 months. Okay, for many of you listening, that's the headline everyone is hoping for from lifestyle studies, right? It sounds incredibly promising for anyone focused on real-world interventions, but my spidey sense for overstatement is tingling a bit here.
I sense it too.
Does the study truly deliver on that promise of meaningful age reversal, or are there crucial caveats we need to consider, especially given that relatively short time frame?
That's precisely the critical question. And it's exactly why we must remain highly skeptical of age reversal claims, especially when they stem from relatively short-term lifestyle studies like this, while the methodology for data collection is, as you said, incredibly impressive.
Yeah, the data set sounds amazing.
It does. 156 women, 7 tissues, multi-omics profiling. It's great. But the core finding that highly compliant intermittent fasting participants showed age-opposing molecular trajectories within just 6 months. Well, that is almost certainly overstated in its implications for true systemic age reversal.
Okay, almost certainly overstated. That's a strong statement. Can you elaborate on why such skepticism is warranted here, despite the robust data collection, despite the seemingly positive molecular shifts? Where should a biotech researcher be applying extra scrutiny?
Certainly. A major point of scrutiny, right off the bat, is that phrase highly compliant. This finding strongly smells like potential post hoc analysis and selection bias. The highly compliant subset was likely identified after the fact, or at least represents an extreme end of the compliance spectrum.
Which might not be typical.
Exactly. It may not be generalizable to the broader population who might try intermittent fasting with, let's say, more average adherence. It's also entirely possible that individuals who are highly compliant with the fasting protocol are also inherently more health conscious in other ways, or that their adherence correlates with other healthy behaviors not fully accounted for, maybe better overall diet quality, more physical activity, lower stress levels. All of these could independently influence molecular markers, making it really difficult to isolate the specific effect of the intermittent fasting itself.
Right, confounding variables.
Definitely. And more fundamentally, 6 months. 6 months is far, far too short a period to claim meaningful age reversal in terms of functional health span or actual longevity. While molecular shifts are absolutely expected with significant lifestyle changes like intermittent fasting or smoking cessation, the body is dynamic, it responds. These changes don't automatically equate to reversing biological age in a functional or clinically significant way.
So we're seeing molecular changes, biomarkers moving in a positive direction, but that's not necessarily the same as reversing the fundamental aging process in a way that truly impacts long-term health span or disease risk.
Precisely. That distinction is absolutely critical. Molecular changes don't necessarily translate directly to functional outcomes or hard endpoints like mortality, disease incidence, or overall health span extension. Think of it this way. If you stop eating junk food and start exercising for a month, your blood pressure might go down, your cholesterol profile might improve.
Sure.
Those are positive molecular and physiological shifts.
Hmm.
But does it mean you've reversed your biological age by several years in just 1 month? Almost certainly not. The link between these observed molecular shifts over 6 months and long-term clinically relevant benefits needs to be robustly established over much longer periods. 6 months just isn't enough time to make that leap.
Okay. What about other factors in lifestyle studies?
Well, we also have to consider expectation bias and the good old placebo effect. These can be really significant in lifestyle interventions. Participants who are highly invested and compliant, like this highly compliant group, might also be highly motivated and expect to feel better or see positive changes. That expectation alone can influence subjective measures and even some physiological responses.
Right, and it's hard to blind someone to intermittent fasting.
Exactly. If you're fasting or not. How were these psychological factors controlled, particularly in that highly compliant group? It's a huge potential confounding factor that's notoriously difficult to mitigate properly in non-blinded lifestyle studies.
That's a common challenge for sure. So while the multimodal data collection is undeniably novel and valuable as a resource, is the core conclusion about age-opposing trajectories really breaking new ground on our fundamental understanding of aging itself, or is the resource the main contribution here?
I argue strongly that the novelty is primarily in the scale and the multi-tissue, multi-omics approach, which creates an incredibly valuable resource for future studies. It's not so much a breakthrough finding on aging itself. This study builds on decades of research showing that lifestyle interventions do affect various vital markers related to health and, by extension, aging. That's not really new.
Right.
For this to be truly impactful for you, the biotech researcher, beyond just providing data to mine, we would need much longer follow-ups, ideally 5 years, maybe longer. Looking at hard clinical endpoints like reduced disease incidence, maintained or improved functional capacity, maybe even mortality data down the line, we'd also need replication in diverse populations. This is only women, predominantly white with robust controls. And critically, a clear delineation of which specific molecular changes are truly predictive of long-term positive outcomes, versus which are just transient shifts related to recent behavior.
So for you, the listener, the big idea here isn't the headline claim of age reversal, which clearly requires immense further validation and skepticism. Instead, the aha moment is the creation of this rich, publicly available data set. It's a resource for future hypothesis generation and biomarker validation. It allows researchers like you to test your novel epigenetic clock algorithms, for instance, against real-world intervention data without needing to run your own costly multi-omics 6-month trial from scratch.
Exactly. That's the real value here.
It's a powerful reminder to always look past the headline, scrutinize the methodology, the claims about specific subgroups, and the data limitations. And to appreciate the real value often lies in the resource generated for future, more definitive work, rather than the initial interpretation.
Precisely. It's a tool for discovery, a data mine for testing new hypotheses and validating potential biomarkers, not a definitive answer on age reversal, at least not yet. Its strength lies in its meticulous multidimensional profiling, which could inform targeted drug development down the line by identifying robust and potentially causal molecular pathways influenced by lifestyle. But that's future work.
Okay. Our final deep dive for today takes us from the lab bench and clinical studies to the more abstract realm of evolutionary theory.
Stepping back to the big picture.
Yeah, this is the kind of paper that truly makes you think about the deepest why of aging. Something that can fundamentally shift our conceptual models, maybe, even if it doesn't immediately impact bench work. The title is Avoidance of Rejuvenation, a stress test for evolutionary theories of aging.
Right. This theoretical paper, coming from the City University of Hong Kong, raises some important fundamental questions about the evolutionary feasibility of rejuvenation. It challenges some long-held assumptions. However, it's crucial to note as we read it that it seems to suffer from somewhat weak empirical grounding in certain areas.
Okay, and where did this one land?
This is also a Bayar-Steff preprint, published just a few days earlier than the others on August 28, 2025. So again, awaiting peer review and published on bioRxiv.
Got it. The authors are Azen, S.I., Lidsky, B.V., and Lidsky, P.V. We'll link it. So What's the central debate this paper is engaging with? What foundational question about aging are they trying to address here?
The paper dives right into the intense and ongoing debate about the biological feasibility of human rejuvenation and what that means for our research strategies. It poses a really crucial fundamental question for the entire field. Should aging research primarily focus on reversing aging in older individuals, essentially trying to fix decades of accumulated damage?
Right.
Or should we instead concentrate on pausing its progression at earlier ages, preventing the damage from occurring or accumulating in the first place? To tackle this big question, they delve deep into evolutionary biology, seeking answers in how nature itself seems to approach aging and potential rejuvenation.
And what do the classical, widely accepted evolutionary theories of aging typically tell us about this dilemma? What's the traditional viewpoint from evolutionary biology?
Well, they review the classic evolutionary theories specifically, damage accumulation, antagonistic pleiotropy, and the disposable soma hypothesis. These theories primarily view aging not as some kind of adaptive, programmed process, but rather as a detrimental byproduct of evolution.
Just something bad that happens late in life.
Pretty much.
Mm.
It's seen as an unavoidable consequence of natural selection prioritizing early life reproduction and survival, with a declining force of selection later in life that fails to effectively remove deleterious mutations or damage that only manifests after the main reproductive period.
Okay.
So from that perspective, rejuvenation, if it were mechanistically possible and not too costly, should confer strong fitness advantages by extending reproductive lifespan and overall survival. Therefore, logically, you'd expect it to evolve quite commonly in species that experience significant aging in the wild.
So, if it's so advantageous from an evolutionary standpoint, and aging is just this byproduct that evolution hasn't bothered to fix properly, why isn't rejuvenation more common in nature?
Yeah.
That seems to lead to a significant paradox, doesn't it? If the classical theories are right.
Exactly. This is the central paradox they identify and build their argument around. The observed rarity of systemic whole organism rejuvenation in nature would, according to these classical theories, strongly imply that it's either mechanistically implausible or just prohibitively costly from an energy standpoint.
Right.
However, and this is a key point for their argument, while rare, rejuvenation does seem to occur in a few species. Think of certain jellyfish, like Turritopsis or planarian flatworms that have incredible regenerative abilities. But paradoxically, in the examples they cite, this rejuvenation is typically induced by environmental stress or injury, rather than being a default, readily utilized mechanism under optimal happy conditions. This seemingly contradictory pattern rejuvenation being potentially beneficial, yet rare, and often stress-induced, is what they hone in on to challenge that classical view of aging as just a byproduct.
That's a truly fascinating observation that nature seems to almost avoid readily accessible rejuvenation, even when it could be beneficial and maybe only triggers it under duress. How do they use this paradox and their specific models to challenge those classical theories of aging?
They use mathematical modeling, specifically looking at lifespan plasticity and highly specialized biological systems like eusocial insects.
Like ants or bees.
Exactly. Ants, bees, termites. In these species, the queen can often live dramatically longer than the workers, sometimes orders of magnitude longer. And sometimes, under certain colony conditions, workers can even revert to a reproductive state, which involves significant physiological changes, maybe even resembling a form of rejuvenation. Their argument, based on modeling these systems, is that this observed pattern, where rejuvenation seems rare across nature, often stress-induced, and not a default optimal strategy for most individuals, cannot be adequately reconciled with the classical theories of aging as just accumulated damage or evolutionary neglect. They claim this reveals an internal contradiction within these long-held evolutionary frameworks, suggesting they are incomplete or maybe even flawed in explaining the full spectrum of aging phenomena we see in nature.
So the existing theories, in their view, can't fully explain why nature largely avoids widespread, easy rejuvenation, even when it seems like it should offer a clear fitness advantage. If those classical models are insufficient, what alternative hypothesis do they propose to resolve this contradiction and offer a new framework for understanding aging?
They propose what they call the pathogen control hypothesis. This hypothesis fundamentally reinterprets aging itself. Instead of seeing it as just a detrimental byproduct or simple wear and tear,
Right.
they propose that aging might actually be an adaptive, programmed process.
Programmed. Like apoptosis, but for the whole organism.
Sort of, yeah, that's the controversial idea.
Yeah.
Their specific argument within this hypothesis is that aging, in this context, might have essentially evolved as a form of pathogen resistance, or a mechanism to control infection spread within a population or a colony.
How would that work?
The idea is, by having a finite, programmed lifespan, individuals eventually remove themselves from the gene pool in the population, potentially limiting the accumulation and transmission of chronic pathogens or parasites that might build up over a long life. They argue that this alternative framework, viewing aging as adaptive and programmed, offers a more consistent evolutionary explanation for understanding why rejuvenation is rare and potentially how we might achieve it by maybe targeting the program itself, linking it to ecological and immunological pressures, rather than just focusing on repairing somatic damage.
Okay, let's unpack this. The idea that aging might be a programmed process rather than just random wear and tear or a side effect of selection that has truly profound implications if it's true.
Huge implications.
It would fundamentally change how we approach interventions. I mean, my old car doesn't get programmed to rust, it just rusts due to accumulated damage and environmental exposure.
Good analogy.
But this paper says maybe our bodies are more complex and aging actually serves some kind of adaptive purpose, like pathogen control. How radical is this idea for most evolutionary biologists? And how does this stack up for us, the biotech researchers looking for actionable insights today?
That's a great analogy, and it really gets to the heart of the ongoing debate. The idea of programmed aging is indeed a radical departure from the prevailing damage accumulation or disposable soma theories, which are much more widely accepted. While it's intellectually interesting, definitely, for those of you who ponder the deepest why behind aging, and it certainly forces a reevaluation of long-held assumptions, it is practically problematic for current intervention strategies.
Why problematic?
Well, the paper's central argument that if aging is just a byproduct, rejuvenation should be common. And then since it's not, maybe aging is programmed, relies on a number of assumptions that are themselves highly debatable within the evolutionary biology community. And the specific pathogen control hypothesis they propose as the driver for this programmed aging, that aging evolved primarily as pathogen resistance, is frankly not well supported by broader empirical evidence across diverse species. While pathogens are undoubtedly a strong selective force, linking the entire complex process of aging directly to pathogen control as its main evolutionary driver is a pretty contentious claim. There's not a lot of direct evidence for that.
Okay. So, while it challenges existing ideas, the proposed alternative might not be the most robust or widely accepted explanation either. What are the specific weaknesses in their argument, particularly for a researcher trying to derive practical meaning from this theoretical paper?
A major weakness, I think, lies in their mathematical modeling. It relies on potentially simplistic assumptions about eusocial insects. As we said, these are highly specialized biological systems. They have unique caste differentiation, often high genetic homogeneity within colonies, very specific ecological pressures. It's really questionable whether findings from these systems generalize well to other species, especially solitary mammals like humans.
Right. We're not ants.
We are definitely not ants. Humans have vastly different life histories, reproductive strategies, genetic diversity, making direct extrapolation highly problematic. Moreover, the examples of rejuvenation in nature they cite are often arguably overstated or poorly understood mechanistically. Or they refer to specific regenerative capacities like a planarian regrowing a head after being cut rather than true systemic whole organism age reversal.
The difference between regeneration and rejuvenation.
Exactly. It's a semantic distinction, but it's a critically important one when we're discussing rejuvenation in a human longevity context. Regenerating a lost part isn't necessarily the same as reversing the biological age of every cell in the existing organism back to a youthful state. They're often not truly reversing the biological age in the way we typically mean it.
That distinction is absolutely key. And stepping back, even if aging didn't evolve to allow for widespread rejuvenation as a default strategy in nature, does that automatically mean we can't engineer it? Evolutionary arguments sometimes feel like they're setting limits on what's technically possible, rather than just describing what happened naturally over millennia.
You've hit on a crucial philosophical and practical point there. Their evolutionary argument about what did or didn't evolve in nature doesn't necessarily constrain what's technically feasible with scientific and medical intervention today or in the future.
Right.
Just because a trait didn't evolve naturally doesn't mean we can't potentially engineer it. We've certainly developed technologies like human flight or deep-sea exploration that defy the natural evolutionary pathways for humans. Our ability now to manipulate biological systems at the molecular level potentially allows us to bypass some of those historical evolutionary constraints. So while it's a fascinating thought experiment, and it might eventually influence how we frame our research questions in the long run. This paper primarily offers, I think, philosophical fodder rather than immediately actionable insights for current rejuvenation strategies or drug discovery. It doesn't provide new targets or direct pathways for intervention or new methodologies for you in the lab this week.
So it's not going to directly impact our current bench research by pointing to a new drug target or a specific pathway we should inhibit or activate?
Not directly, no.
But it does challenge fundamental assumptions about aging's origins. Is that a model change in a sense, even if it's not an immediate therapeutic one?
Yes, I think you could call it a different kind of model change, a theoretical one maybe, and the deepest conceptual level about the why of aging. Not one that immediately changes how you design your experiments tomorrow or how you screen for new drugs today. It asks us to question our foundational understanding of why aging exists at all, which over time can subtly influence the kinds of questions we ask and the types of interventions we consider, perhaps pushing us towards novel approaches if, say, the programmed hypothesis ever gains more robust empirical support. Right, if that evidence appears. Exactly. But for now, for you, the biotech researcher, it doesn't immediately point us down new therapeutic avenues. The big idea here, really, is that while classical evolutionary theories suggest rejuvenation should maybe be common if aging is merely accumulated damage, its rarity in nature might hint at a deeper, perhaps adaptive purpose for aging. This challenges our fundamental models, even if the specific pathogen control mechanism they propose still needs substantial empirical validation. It's a question mark, not an answer.
Okay, so reflecting on the week, what have we learned from this batch of papers and maybe the broader trends shaping the longevity landscape that we touched on with ARDD? Well, we didn't uncover any earth-shattering breakthroughs that demand a complete reevaluation of your research direction right now.
Right, no bombshells this week.
No bombshells.
But we did see a promising yet clearly preliminary study on Yamanaka factors
Yeah.
and vision restoration in mice. It highlights the potential of targeted reprogramming and the importance of identifying specific downstream effectors like GSTA4, even if it's definitely an incremental step needing lots of validation. We also looked at that massive data set on lifestyle interventions from the Tyrol-Giesende Atlas, which, while methodologically impressive and a fantastic resource for the community,
Yeah, the data set itself is great.
it serves as a stark reminder that age reversal headlines from short-term lifestyle studies require extremely careful scrutiny, particularly regarding compliance claims and short timeframes.
Right. Skepticism required.
Highly required. And finally, we dove into that theoretical discussion, challenging our foundational understanding of why we age at all, prompting us to maybe consider alternative evolutionary frameworks, even if they lack immediate practical application. And woven into that was the truly fascinating observation from ARDD, the conference's apparent willingness to embrace more audacious long-term concepts like full-body replacement, which signals perhaps a broader conceptual shift in the field's long-term vision beyond just drugs.
Yeah, it's a week that really underscores the complex journey of scientific discovery, doesn't it? Lots of small, precise steps forward at the bench, a few intriguing detours into fundamental theory, and then the occasional bold, almost sci-fi prediction from a conference stage that truly widens the scope of what's considered possible maybe in the distant future. It feels like a field constantly pushing boundaries in every direction.
It really does. And for you, the biotech researcher listening, it reinforces that crucial balance between recognizing genuine scientific progress, appreciating the value of new data resources like that atlas, but always maintaining a vigilant, critical eye on the claims being made, particularly those promising rapid age reversal or simple solutions. Every week, we're just building the puzzle piece by piece, often slowly, even when a full, clear picture feels very far off. That careful analysis of methodology, the controls, the data limitations, that's what truly drives robust science forward.
Absolutely. And it's in those details, the rigorous methodology, the tight controls, the demand for long-term data, where the true value and the potential for future genuinely model-changing work really lies. That's where you, the sharp-eyed researcher, make your most significant contribution.
So maybe a final thought to leave you with. Given our wide-ranging discussion today, considering the scope from targeted cellular reprogramming with things like GSTA4 all the way to the audacious concept of full-body replacement, and even questioning the deep evolutionary roots of aging itself, if the most profound breakthroughs often come from challenging existing models, What fundamental assumption in longevity research are we, collectively as a field, perhaps overlooking or maybe holding on to just a bit too tightly right now? An assumption that, if questioned more deeply, could truly unlock the next paradigm shift.
That is definitely something for you to mull over in the lab this week. Thank you for joining us for this look into the latest longevity papers. We'll be back next week with more.